video, we're going to be talking about the biology of memory. And I think this is something that is super cool because it's really neat how the biology works, and also something very relevant too as you try to pack all these facts into your head for the MCAT. So for the sake of this, I'm going to divide memory into 3 different parts. So I'm going to talk about working memory. Okay, so that's if you need to remember something for a few seconds, and then short-term memory. Okay, and that's going to be like if you need to remember something for a few minutes or a few hours, so a little longer than working memory. And then, of course, what we want for the MCAT exam is long-term memory. So that's a few hours to the rest of your life in terms of temporal scale. And the reason that these are all different types of memory is because they actually have different biology underlying them. So I'm going to go talk about each one in turn. And let's go ahead and start with working memory. So in the case of working memory, you're going to have a network of neurons, but I'm just going to draw 2 for the sake of this. So I'm going to have 1 neuron here, that's neuron 1, and then I'm going to have 1 neuron here. And let's put some concepts to these, just so it's a little easier to intuit. So let's say this first neuron is the neuron that is activated when you see your friend Tony's cat. Okay? So this is your Tony's cat neuron. And then this neuron over here is going to be your sandwich neuron. So this is going to fire whenever you see, feel, or touch a sandwich. And so let's say, for the example of working memory, you see Tony's cat eating a sandwich, and you see Tony in the distance, and you're just going to remember that Tony's cat was eating a sandwich for a few seconds until you see Tony and can tell him, but you don't necessarily want to remember that Tony's cat was eating a sandwich for the rest of your life. So in this case, you're just going to have activation of both of these neurons. So remember, activation of neurons is caused by depolarization, so lightning bolts. And this depolarization is what's allowing these neurons to communicate with one another and to signal, in this case, the concept Tony's cat or sandwich. So in this case, you're going to have Tony's cat neuron firing and sandwich neuron firing, but you don't really need to create any lasting changes in the relationship between these neurons because you're only going to have to hold it in your head for a few seconds. So, so these are just going to be firing independently. So working memory, the biological basis of this is basically network activity. So I showed 2 neurons in the network here, but maybe there's also, you know, the texture of the sandwich and the context in which the cat ate the sandwich and why you need to tell Tony about it. And so there's going to be a bunch of neurons here that are all independently firing with these different disparate concepts. Okay, but for short-term and long-term memories, we're actually going to need to create changes in the relationship between these neurons. And remember, neurons connect and talk to each other at synapses, so you're going to have synaptic changes. Okay, so for this, let's zoom in. Let's see. Draw a synapse there. Okay, and because this is very small, let's go ahead and zoom in on this. And I'm just going to move over a little bit so we have some room. Okay, so let me draw that out a little larger. Okay, so here we have the synapse between Tony's cat neuron and sandwich neuron. Okay, and I'm just going to draw sort of the essential components of the synapse here. So Tony's cat neuron is going to be our presynaptic neuron, so it's going to have these synaptic vesicles, which are filled with neurotransmitter, and that's going to be its means of communicating. And in this case, they're going to be filled with glutamate, which is true for 90% of neurons in the brain, so pretty common. And then the postsynaptic cell has to have a way to respond to that, and that's going to be with receptors on the surface that recognize glutamate and transduce the signal of glutamate into the cell. Okay, and we're going to have 2 different kinds of receptors on this neuron, which are really important. So pink and blue. Okay. So before I move on, let's talk about the properties of these different receptors, and that's going to help us understand how these synaptic changes come about. So I'm just going to zoom out a little bit here so we have some room to talk about them. Okay, So these pink ones are going to be called AMPA receptors. And don't worry too much about the names. What's going to be important is sort of the differences between these receptors. Okay, and then the blue ones are called NMDA receptors. Okay, and so what are the important aspects of receptors? Okay, so first, if you're going to describe a receptor, you want to describe what activates it. So what's going to cause it to open and do its thing, and what effect does it have on the postsynaptic cell when you activate it. Okay, so in the case of AMP receptors, what activates it is glutamate, so it's a glutamate receptor, and it's pretty simple, so that's it. Okay, for the NMDA receptor though, it's going to require glutamate because it's a glutamatergic receptor, but it's also going to require depolarization of the postsynaptic cell, so I'm going to say plus lightning bolt. Okay, so this is going to be the key difference here. And then in terms of effect on the cell, AMPA is going to open up and allow sodium ions into the cell, so you're going to get an increase in sodium inside the cell. NMDA receptors are also going to let sodium inside the cell, but they have another added effect, which is allowing calcium in the cell. Okay, so also calcium. There we go, 2 plus. All right, so that's going to be our key differences here. So now let's look, knowing these properties of the receptor, what's going to happen here. So the first time you see Tony's cat, you've never seen it before with a sandwich, then you're going to have glutamate release from the Tony's cat neuron here. So you're going to have glutamate out here. And you have these NMDA receptors at the synapse. So what's going to happen in the sandwich neuron? So you have glutamate, so that's good. But you don't have depolarization of this postsynaptic neuron. So the glutamate is going to bind, but these NMDA receptors aren't going to open because they don't have the depolarization that they require. Okay, so how are you ever going to get the sandwich neuron to respond to the Tony's cat neuron? Well, when you see Tony's cat and a sandwich at the same time, then you're going to get glutamate release from the Tony's cat neuron. And because you see a sandwich, you're going to get depolarization in this neuron. And those are both requirements for NMDA receptors. So when you see Tony's cat eating a sandwich, you're going to get calcium enter through these NMDA receptors. And this calcium is going to do a couple of things. So the first thing the calcium is going to do is come bind to these AMPA receptors, which are hanging out here, and cause them to actually insert in the membrane. So, okay, so let's look at what effect that has on this communication. So imagine next time you see Tony's cat, you have these AMPA receptors in the membrane. So you have glutamate release from the Tony's cat neuron, and AMPA only requires glutamate. So you're going to get activation of the sandwich neuron when you see Tony's cat. You're going to think about it eating a sandwich. So that's perfect. But these AMPA receptors are only going to stay in the membrane for a few minutes. There are a lot of things that come and take them back out of the membrane. So this is going to work for a few minutes or a few hours while these receptors hang out. But if you want to remember this long term, this calcium is going to have other effects. And that's actually going to be through protein synthesis. So in this case, calcium is actually going to go back to the cell body, and we can look at that over here on our full neuron pictures. So calcium on the sandwich neuron is going to travel back to the cell body and activate proteins which can bind to DNA and actually cause transcription of new proteins. And so this is going to mediate the really long-term effects, even after those AMPA receptors have come out of the membrane. So when we're looking back at our diagram here, we have working memory is just network activity. Short-term memory and long-term memory both require synaptic changes and through some of the same machinery. But specifically, short-term memory is going to require receptor insertion, while long-term memory is going to require new protein synthesis. So you can see, a lot of biology is common to these processes, but some of it isn't. And that's sort of why we have separated these psychologically into different types of memories, because they have different biology behind them. So hopefully that gives you sort of a way to intuit how memory is happening, and good luck. I hope you get lots of AMPA receptor insertion and new protein synthesis to help you with this MCAT studying.